Okay, sorry about that. I don't know
No,
what happened
no, I think
exactly.
it's perfectly fine. Venkatesh,
Yes, sir.
so we can quickly start.
Yeah. Good evening and welcome you all to the fourth talk of Bangalore India Nanotech 2022. The Vision Group on Nanotechnology Government of Karnataka under the honorary chairmanship of Professor Bharat Ratna Professor C.N. Rao and the chairmanship of Professor Navakat Bhatt has initiated this webinar series. This is the fourth Nano Talk in this series. The guest speaker of today's Nano Talk is Dr. Shantikumar Nair, Dean of Research, Amrita Vishwa Vidyapeetham, and Director School of Nanosciences and Nanomedicine, Kochi. Topic of this today's talk is Nanoscience and Molecular Medicine, Current Trends and Future Prospects. I welcome you, sir.
Thank you.
Now I request Professor Anindya Das, Professor, Indian Institute of Science and convener of Bangalore India Nano monthly webinar series to introduce our speaker of today's speaker.
Yeah, thank you, Venkatesh. Without taking too much time, it's my great pleasure to introduce today's speaker, Professor Shantikumar Nair, who is eminent and renowned nanoscientist and has contributed immensely in the area of molecular medicine. Professor Nair currently Dean at Amrita Vidyapeetham and Director of Amrita School of Nanoscience and Molecular Medicines. To quickly tell his career and his achievements, Dr. Nair did his Bachelor of Technology from IIT Mumbai in 1976, followed by he did his Master of Science in Material Science and Engineering from Columbia University, New York in 1978, and did his PhD in Material Science and Engineering from Columbia University in 1983. He joined as a senior research scientist in Columbia University from 1983 to 1985, then joined as a professor in material science at University of Massachusetts, Amherst. Dr. Nair is recipient of many prestigious awards. To name few of them: Presidential Young Investigator Award 1988 from President Ronald Reagan, USA. DST National Research Award 2011, MRSI Gold Medal 2012, Professor C.N. Rao India Nanoscience Award 2014. Professor Nair has published more than 600 journal articles, 3 books, 35 granted patents. His H index is more than 80. I think with this, I'll hand over to Professor Nair. And today he is going to deliver talk on nanoscience and molecular medicines, current trends and future prospects. Professor Nair, stage is yours.
Yeah, thank you. Thank you, everyone. First of all, thank you very much for inviting me to this very special and important meeting. Anything that Professor Rao does immediately has national importance. And I'm really honored to be invited to this event to deliver lecture to your audience. It is especially, honoring to talk about all the work that has happened in nanosciences and molecular medicine, to give you a bit of an overview, because, for the last 15 years, we've been working on it, step by step and making a lot of progress. And the field has also grown enormously and has become so important. So it's an honor indeed to give a little flavor and an overview of the field to your audience. So I have a lot of slides, but I'm not going to spend a lot of time on every slide. In case there are questions, I can come back to some of the detailed slides and talk about specifics. But I'll keep it slightly broad so that you can get a flavor for what is going on in nanosciences and molecular medicine. First of all, I want to mention that when I say nanosciences and molecular medicine, they're not really two fields. They are very commingled together, and the one affects the other. And this synergy between the two is really the reason why the field has become so important. So with that, let me say a few words about the Institute. I don't want to spend too much time on that. That's not the purpose of this talk. Our institute is now an 8-campus university in several states. We have done very well in rankings, and currently we are as institution of eminence under the government of India. So that has happened in part because of the emphasis on research at our university. And we have at our Center for Nanosciences, I myself cannot see some of these slides, a lot of accomplishments, with Professor Rao's support and help and the support of DST, we started in 2006. And we've come a long way in the last 16 years. We were the first nanobio center, nanobiomedical center in India. And with DST support and Professor Rao's blessings, we got a lot of things that we were able to accomplish a lot, and it's been a wonderful journey for us. Let me see. So primarily what I want to say is that the area of molecular medicine and nanotechnology and nanomedicine is very interdisciplinary. It cannot be tied down to any particular field like physics or chemistry or engineering. And for the most part, it is translational, which means that it goes from the lab to the clinic, or you could say lab to society, to products that helps people. And that's a very important part of nanosciences that has endeared itself in the field of practical applications and to people and to society in general. And it has the potential to make medicines more effective, less expensive, unlike many new technologies, which end up being substantially more expensive. So these three aspects are very unique to nanomedicine and molecular medicine. And the interdisciplinary aspect, if you just look at some of the interdisciplinary aspects, there's material science, there's medicine, there's engineering, there's biology, there's chemistry, there's physics. It is a very, very interdisciplinary program. On top of it, there's a lot of engineering involved with mathematics and bio and data analytics, computer sciences like in bioinformatics, which is a very integral part now of molecular medicine. So the field is very exciting, and a lot of students find it very interesting to have that kind of exposure. And I mentioned translation. When you're talking translational, it's very important to point out that there are a lot of regulatory issues, scale-up issues, commercialization issues, which a lot of people in the field of nanomedicine are dealing with. In fact, I was part of the DBT Nanotechnology Policy Committee, looking at aspects of how do you actually commercialize nanomedicines in India. So these are all important questions that are being addressed or have been addressed, but it shows the tremendous translational potential of nanomaterials. Now, all of you know very briefly, and some of the audience here may be students or postdocs, or maybe someone who's not so used or so familiar with the essential idea of nanomaterials. So when we talk about nanomaterials, what we are talking about is a very high surface to volume ratio. That itself is not necessarily so unique. You have a surface to volume ratio that's inversely proportional to R, which means that when you go to a sufficiently small size, you get very surface active behavior. It's surface controlled. And both biology, engineering, chemistry, physics is all very strongly related to surface properties and surface behavior. And so the field of nanomaterials, if you go forward, if you look at that graph on the right-hand side, the surface area, if we go below 100 nanometers, the surface area just explodes. In fact, if you look at metal oxide materials, you have surface area as high as 7,000 meters squared per single gram. So it's going to be extreme. These are metal oxide framework materials, and those materials are highly surface active. And in biology, surface activity is extremely important. So this is one important reasons why nanomedicine and nanomaterials have taken center stage in the new field of nanomedicine. And if you look at the history of nanomaterials, in 1990, back when I was in the US, the work at Cornell University, modernized nanomaterials with the introduction of layered silica clay nanoplates. And a lot of work was done at that time to introduce these materials into our current conventional micro scale and larger materials. And that spawned the field of nanotechnology. And very quickly, these sorts of materials were recognized as very important in medical field. In fact, Doxil was first nanomedicine that was released in the US for breast cancer in 1995. And after that, a lot of nanomedicines have not only been released, they've been approved, FDA approved, used. So nanomedicine is not an idea anymore. It is actually a practical, useful, and approved approach to improve healthcare. And if we come to India, closer to home, 2007 is about the time that India started producing nanodrugs. Originally, Nanoxel was produced by Dabur and subsequently manufactured by Fresenius Kabi India Private Limited. And it was a unique material, especially because of the way that it was processed. It was castor oil free, chemophore free, and therefore less toxic. And so it had a very good market and it was fairly successful. It competed with Abraxane, which is another material that was used that was similar to Dabur's product. And in 2004, as you all very well know, graphene was discovered, which is a two-dimensional material with very, very high surface area. And we were one of the first groups that showed that graphene is biodegradable. And if that is so, it may not be so toxic to deliver drugs with the graphene. You can functionize graphene with drugs and deliver it into the body. And so that also opened up the field. Subsequently, there's a lot of work on the use of graphene in medicine. And in 2014, Sun Pharma released a new material. These are innovative materials. These are not generics that were essentially duplicating other nanomedicine, but new material, which is also castor oil and chemophore free material. So we are now in the process with a lot of support from DBT to generate a new class of oral nanomedicines. As you know, a lot of the nanomedicines that have come out in the past have been IV nanomaterials or somewhat more invasive ways of delivering drugs to the body. But if we could deliver nanodrugs orally, that is going to be a game changer. And so we have developed a platform, and there's companies interested in it, and we are trying to go through regulatory approval and DCG approval, where these oral nanomedicines can have substantially high bioavailability, as well as into the bloodstream as nanoparticles containing drugs. So this is not an easy task, and it substantially short up the bioavailability of drugs and also the ability to target particular cells or molecular markers through the oral route. And I think that is going to be a game changer. And we hope that it will really help patients who can take drugs from home. So instead of coming to the hospital for an IV treatment. So now a lot of work is now going on, as I will briefly talk about during the course of the talk, on cell therapy and immunotherapy. You know that conventional synthetic drugs and chemo drugs, while useful, has a lot of toxic side effects. And to be able to use the body's own defenses, such as CAR T cell therapy or other immunotherapies or checkpoint blockers, etc., we have a way to treat diseases much more effectively and with much less toxicity. And nanomaterials and nano medicines are poised to enter in a big way into cell therapy and immunotherapies that are more effective than conventional or current cell therapies and immunotherapies. And I think that this potential for nanomedicines to enter into biologics is going to be another game changer. And so we are all looking forward to it and trying to stay, with the research that is happening globally. And the idea with all of these is targeted treatment. That is molecularly targeted treatment, which can be delivered with the help of nanomaterials so that you have very effective and very, very little off-target effects. So that's kind of the overview of where things are going. And now I'll just go through a few examples of some of the things that are being done. And so to quickly recap, you can deliver using nanoparticles. Usually that has been done through IV, but as I said, you can do it orally now with the new platforms that we are developing. But you can also, and with these nanoparticles, the beauty of it is you can use multiple drugs using core-shell systems. We are one of the first to have a core-shell patent on a multi-drug, core-shell nanomedicine for oncology. And it has some very beautiful plus points, which you really cannot achieve by separately giving multiple drugs, which has their own, kinetics, in the pharmacokinetics in the blood, so that there are some significant advantages. You can also add, and as I'll show you, contrast agents so you can see where they're going. The second type is nano implants. Now, instead of delivering nanoparticles, even either orally or IV, you can actually implant them. One of the advantages of implants is that you can deliver drugs for months. And this is a very significant advantage for many sorts of things, even for simple things like, let's say you want to deliver testosterone to some person, which is another important therapy. It's oftentimes when you get older. So you can deliver testosterone for 1 to 2 months using nanosystems that contain testosterone. You can use sheets containing chemo drugs that can be peritoneally implanted and can deliver drugs for 3 months. So you don't have to continuously infuse the drug into the system, cause so much of problems for patients having to constantly visit the hospital. You can have wafers that you can implant strategically where you need it, and it will deliver drug for a long time. For example, for brain tumors, we have developed some wafers that can release drug for a long time. We have gels that you can use that release drugs for a long time, and they can be locally injected or subcutaneously injected. And you can have other solid implants as well, which can be used. And the point I want to make is that we have so much of flexibility to control the pharmacokinetics. Every single way that you deliver drugs changes the pharmacokinetics of how the drug is delivered. It improves the local action. It can be more targeted. So the traditional way of either IV or oral is one way, but there are substantially different ways that one can treat depending upon the problem using implants. So I'm not going to touch too much on it except to say that the shape of the nanomaterial also matters. If you have a nanomaterial that is like a bullet shape, as opposed to a circular one, it enters, whether it is through the cell or through the skin, in a substantially more enhanced fashion. And so you can control the shape of the nanoparticle and affect the uptake of the drug. So we have done a lot of work on how the shape affects it, what particular types of shapes are useful, and so on. Another aspect I want to touch on, and I spend just a minute on this slide, is if you compare conventional medicines and nanomedicines, you see fundamentally a very stark contrast. If you look at the blue curve, which is the curve for the conventional medicines, you can see that the efficacy increases. And as the efficacy increases, the toxicity increases, and eventually it reaches a plateau in the efficacy. So you cannot really increase the efficacy beyond a certain point, but the toxicity keeps on improving if you increase the dose. So I call this efficacy limited behavior. This has always been a problem with many conventional drugs and conventional medicine because they're efficacy limited. Now, a paradigm shift happened when we went into nanomedicines that are molecularly targeted. And that is that you can see that as you increase the efficacy, there is a decrease in toxicity. And you can actually, or if you go the other way, as you increase, you get a material which is at high efficacy, but the toxicity is limited. So you get a material that is toxicity limited rather than efficacy limited. What does that mean? It means that the nanomedicine has much significantly lesser toxicity, but better efficacy. And this is something that has given a lot of impetus for the development of nanomedicine because it's not efficacy limited, unlike conventional medicines. So the other part is that they are much smaller than cells, so they can enter cells and they can start affecting the cellular pathways inside the cell. So intracellular pathways can be affected. They can go into the nucleus. You can affect the cell nucleus. You can tinker with the DNA. You can deliver genes. You can do gene therapy. You can, as we very well saw in the COVID era, the use of mRNA vaccines, you can deliver mRNA through nanomaterials efficiently. So any which way that you can enter drugs efficiently into a cell is a very powerful tool to have to increase the efficacy. And you can also use nanomaterials to edit the nuclear DNA through, for example, CRISPR-Cas. So you can, and we have done that. We have encapsulated CRISPR-Cas protein into a nanomaterial and found that you get fairly efficient gene editing using nanomaterials, which means that they get into the cytoplasm, get into the nucleus, and they're able to accomplish gene editing as well. You can also accomplish CAR T cell, modification of T cells with nanomaterials efficiently compared to viral-based vectors. So why can we do all this? Because nanomaterials can efficiently enter a cell, they can affect the cellular pathways and enter the nucleus. And of course, all of this requires you to design the nanomaterial chemistry, the charge, the size, the shape, and all of that. But that's part of what we all do these days. And we are able to control things so that we can improve the efficacy. In fact, when we started using nanomaterials for gene editing, initially didn't really do much of anything until we controlled all these parameters. So the aspects that I don't have time to get into is all the aspects of how we need to control the nanomedicine by the surface, the shape, the chemistry, the charge are all extremely important in terms of how well it gets into the material, how well it gets into the nucleus, what is the extent of drug loading, the extent of drug release. You want the drug loading to be high and you want the drug release to be good when it's inside the cell. So all of these aspects can be modulated and controlled. And that is what makes nanomaterials a very powerful candidate for various applications. And you can target the nanomaterial by various features that you can functionalize to the nanomaterial that allows you to target whatever you want to target in. If you know what is the molecular target for the disease, you can attach the appropriate ligand to the nanoparticle and you'll get more efficient uptake. So all of that can be done in case of nanomaterials. You can block receptors, you can get uptakes increased. It has been used very successfully in immunotherapy. It has been used successfully in a variety of applications, even many that we have done and that we are planning to translate to the clinic in the near future. So the generic idea is that it's the nanomedicine is a device. Really speaking, it's a device. You can have materials in there that we can call optical trackers or radiation trackers so that you can know where the nanomaterial is going through the contrast that it provides. You can put drugs in there, proteins in there, biologics in there, mRNA. We have done siRNA delivery for gene silencing and so on. You can put whatever it is that you want properly functionalized into the material, and then you can have targeting ligands, and you can also have other components like PEG, which can increase the circulation time so that the nanoparticle is not immediately cleared by the system. So it's really a device in some ways when you look at it, but as well as the regulatory bodies are concerned, it's not treated as a device, it's treated as a medicine. As long as you have some drug in an engineered system, it's a medicine. So from a regulatory standpoint, we are still treating nanomedicines as medicines, not as devices. But I'm just pointing out that in some ways, it is actually also technically and strictly a device because it has a complicated series of multiple functions, which usually devices have. As an example, we developed protein nanomedicines, which contains gold nanoclusters. And gold nanoclusters have very significant red glow contrast to it. And it can be used to, for example, diagnose a disease. For example, leukemia. If you look at leukemia and you can see the red glow that comes when you put gold nanoclusters into the protein material, and you can see that you can functionalize it with an appropriate antibody. In this case, we functionalized it. It was a CD33 conjugated gold clusters that were encapsulated within protein, and it was functionalized so that it targeted leukemic cells. And you can see that AML leukemic cells were very easily and quickly identified when these nanoparticles accumulated around the surface due to the CD33 targeting. And because of their luminous red color, visually you could immediately tell that the patient has leukemic cells there. So you can see that it's a very simple color method of being able to diagnose diseases very quickly. And that's a tremendous advantage. Another aspect of what we found is that we were able to show that in these materials, there was, especially in CML, transferrin expression is very high on the surface of the cells because these cells try to get iron out of the bloodstream, and so it overexpresses transferrin. So one way to deliver drugs to CML patients is to have transferrin targeting. So if we now use a core shell material where we use a material inside which contains the drug, and outside we use transferrin protein, then essentially I'll go back to that. It automatically targets the CML leukemic cells, delivers the drug because it targets the transferrin receptors expressed on the cell surface. So the transferrin protein that is contained on the outside of the nanomedicine targets the transferrin receptors, and it's able to deliver drugs more effectively. And if you see this core shell system way on the right side shows basically substantially more cell killing than all the other methods. If you look at sorafenib, which is one of the drugs, or imatinib, or there are other versions or generations of it, lenvatinib, for example, and so on. They are, if you give the free drug, it's hard, it's relatively less effective. But if you're able to target it, especially using the core shell system to the transferrin, you get extremely high efficacy. So I wanted to show this to illustrate that you get very good targeting and increase in efficacy of the nanomedicine by this molecular medicine approach, a molecular targeting to the particular disease receptors. And you can see how well, you can even see it in SEM, this core shell system. And we have now taken this material through DBT funding, through, through a GMP processing, which is for scale up and commercialization. And we hope that this sorafenib-based material, which has ability to be absorbed efficaciously, will have tremendous value. And it doesn't only have value in in CML, but also in liver tumors, where sorafenib is used in late-stage liver cancers. So we are, with the DBT support and funding, we are scaling this up using hypershow homogenization techniques to create uniform particle sizes in the GMP. So we have a GMP set up and established at Amrita for nanomedicines, which is one of the first in the academic area in the field of nanomedicines. And we are hoping to take this product to the market. So the idea, and which I don't have too much time to get into, is that we have now a technology base to increase the bioavailability. So instead of giving things IV, which I talked about earlier in the talk, we can now have a way to increase the bioavailability very substantially. So what we need in the end that I've shown is we need a nanoparticle that has mucoadhesive layers and a mucopenetration layer so that it goes through the intestinal walls and gets into the bloodstream as nanomaterials, as nanoparticles. And we have been able to show that using TEM, and we have seen nanoparticles that are in the liver after oral treatment, not as free drug, but as drug encapsulated nanoparticles. So this idea that you can actually, so to speak, inject nanomedicines into the bloodstream orally is a very powerful concept. And it, I feel, is going to be a game changer in increasing the bioavailability. In fact, if you go to the bioavailability profile, this is not the latest data. Latest data is even better than this. You get a bioavailability increase in a factor of 3. That's a huge increase in bioavailability. What that means is that now we don't just simply have to talk about sorafenib or liver tumor. Pretty much anything where you need very high bioavailability, or we are looking at drugs with very poor bioavailability, can now be injected into the system through an oral nanomedicine route. Which is something that we are now currently exploring and developing in concert with a company. And we hope to have a platform. It'll be the first such platform where we can get oral nanomedicines in nano form absorbed by the body. And so we are excited about that, and we are continuing to work on that. So now, very quickly, diagnostics, just as a little break from nanomedicines and therapy, we'll just talk a bit about diagnostics, especially light-based diagnostics. And that's a very useful way. By shining light on a tumor, can you tell whether the tumor is cancer or not? That's a very, very useful tool, not just in skin tumors, but it could be oral cancer. It could also, the light could also be shown through fiber optic that is inserted into the body and shown on to potential tumors. How does it work? You collect the Raman signal, and then you basically deconvolute the data using various artificial intelligence-based approaches, and you can tell whether it is a normal cell, whether it is, this is a high-grade cell, whether it is a normal tissue, in which case it shows up in this box. If it is low-grade tumor, it shows up in this box, and there is a significant amount of difference. And all that we are doing is basically bouncing off a laser beam from the surface of the cancer using our own tool, which has a very fine distributed roughened nanoscale surface on it. And that enhances the Raman signal enough to get very high sensitivity. And we can distinguish not only whether it is cancerous or non-cancerous, but we can also distinguish whether the material, what type or what grade of tumor is this. That actually is a very useful thing. So nanomaterials is not only in drug delivery, but it is also in medical diagnostics, a very useful approach in medicine. And so there's a lot of technology integration in this. The laser work required a lot of physics students working on it and deconvoluting the data. We have been working on a lot of work where we're combining it with molecular medicine, by which I mean, what are the molecular targets? Once you know what the molecular targets are, then we can combine a nanomedicine or design the nanomaterials for those particular targets. And that's essentially what nanomedicines actually do. Another part of nanomedicine, which I didn't talk about so far, is metronomic therapy, which means that instead of delivering drugs, fairly suddenly, we are delivering a small amount of drugs over a longer period of time. So it turns out that if you are continuously delivering the drug over a longer period of time, the therapeutic value is substantially enhanced. We haven't fully understood the mechanisms of that. If we give less drugs over a longer period, rather than more drugs over a shorter period, the first one works very much better. And so the question is, how less of a drug can we use to give metronomic therapy? If the drug level that you have to use in metronomic therapy is very less, it's a huge advantage, because then you can use the concept of metronomic therapy with nanomedicines to give far less than the toxic side effect levels. But at the same time, the very act of giving metronomic therapy improves efficacy. It's especially true when we are looking at immunotherapy, and we're doing some work with Northeastern University looking at how metronomic therapy works in immunotherapy scenarios. And it changes the molecular markers, the expression level of the cells. So the biology of the disease changes in metronomic therapy compared to non-metronomic therapy. It's not that simply that you're giving less drugs over a long period of time. You're changing the biology of the disease. And this is something that is a new frontier, which has a lot of potential and is very exciting. And we are very much interested in understanding how metronomic therapy changes the biology of disease. And of course, another aspect is longer circulation, longer half-life, and so on, which I think is quite understood. But the concept of metronomic therapy is quite interesting. So now you also, I think, many of you know, you've heard about personalized therapy. Primarily, it is looking at the molecular characteristics of the patient and developing the drugs for that. The molecular characteristic of the patient controls the efficacy of the drug, what drug is used, how it is targeted, and so on. So nanomedicines are perfect for metronomic therapy. And I showed you nanoscale diagnostics. Give you an example of, again, how you can use nanomaterials in various tumors by combining different therapies. Now, one of the things that we do even today, we use radiation with chemo, right? And we are actually already combining treatments in various therapies. But you can do that in much more effective ways, I think, when you're able to create a multi-therapeutic nanoparticle, which is capable of providing that combinatorial therapy. So you can use, this is an example where we use PDT, which is photodynamic therapy, along with chemo, radiation, and so on, in brain tumor. In brain tumor, chemo therapy and radiation therapy is a standard of care. However, the problem with it is it never works. People always die after brain tumors are detected within a year because of the recurrence of the tumor. And we need to combine chemo and radiation with the standard of care with something a little bit more effective at the local level so that you prevent the recurrence of the tumor. And that's what we have been working on. This is an example where we use light photodynamic therapy to combine, and we found that we can get substantial improvement. So once you resect the tumor, you shine light on it, you have a photodynamic drug that is placed there, and you shine light on it, it removes all the residual cancer cells, and it couples with chemoradiation to give a far better effect on overall patient outcome. And the idea that I wanted to show you here, you all know about photodynamic therapy, but photodynamic therapy with nanoparticles is a much better ballgame. Because if you look at it, I'll just show you very quickly. We call it Nano Photomedicine Gel. So we make a nano photomedicine instead of just a photomedicine. And the idea is, if you look at the black curve, which is the nano photomedicine, it gives much, much higher enhanced signal. And in fact, if you look at, I'm just going to skip a few things, the absorption of laser light by the gel gives rise to far more singlet oxygen species, which causes ROS cell death of the tumor compared to the naked drug. Why is that? If you look at the singlet oxygen emission, it peaks and is substantially higher than that for the free gel. So the nanomedicine, by putting the photodynamic particle inside a nanoparticle, photodynamic drug inside a nanoparticle, gives rise to much more efficient absorption of the laser and much more efficient release of ROS species. And that's what makes it so very effective. And I'm going to skip through many of these slides. You can see that the NPM gel shown on the bottom causes a lot of cell death compared to the free drug. And in the cell death, you can see the rightmost curve, all the cells die with the nanophotomedicine, but not with the free photomedicine. And if you look at the curve, say this curve here with the cell viability, yellow is the nanophotomedicine. You can see that even MGMT positive cells, which are resistant to chemo, is fully sensitive to the nanophotomedicine. And so you have ways to do combinatorial therapy very effectively. And I'll skip some of these slides now. And we've also developed in a gel form so that we can basically, that gel contains even a chemo drug. Even the chemo can be delivered once you remove the tumor and prevent recurrence of the brain tumor. So we have done all the large animal studies in pigs and so on. We have come quite a long ways away in terms of trying to translate this. And we use fairly low fluence, 20 joules, 10 minutes. And we can see that when after 60 days, the only case where the animals are tumor free is in the gel treated animals containing the nanophotomedicine. Okay. So, and we've done all the toxicity work and so on. And we've also developed many other contrast nanomaterials. I didn't have a chance to talk about contrast nanomaterials because of time limitation. This is a nano calcium phosphate iron contrast material. And the beautiful part of it is that calcium phosphate is highly biocompatible. It is easily absorbed in the liver, and the iron gives rise to dark contrast. And so if you look here, it is absorbed around in the surround. If you look at liver cirrhotic lesions, the iron nanoparticles are absorbed all around it because of the macrophages that are present there. And all the cirrhotic lesions show up as light dots very, very clearly after NCP iron injection. It's extremely, our clinicians are very excited that they could identify liver cirrhotic lesions at a very early stage. And so contrast nanomaterials are very, very important. And so we've done that and published it also. Now I talked about combinatorial treatment, what we have done, and I'll go straight to, I'll skip the introductory parts and go straight to what we have done here. I'm going to, two things. One is RF, which is radio frequency ablation. So if you use nanoparticles for radio frequency ablation, you can ablate a much larger size of tumor than is currently possible because it spreads very nicely within the tumor. That's one. The second thing, and you can see that the localized temperatures is also much better because it's absorbed in the tumor wells surrounding the tumor. The temperatures are less, and the area that it affects is also very high. Now what we have done is combine radiofrequency ablation with basically radio embolization, which means it's basically like technetium 90, which is used to release radioactive radiation into the tumor. So it absorbs, it embolizes the arteries near the tumor. It basically radiates that tumor, and at the same time, it contains nanomaterials which contain chemo drugs and which are RF sensitive. So you can, with one nanomedicine, you can do RF ablation, radio embolization, and chemotherapy. So these are not possibilities that can be done very effectively in a single go with such precision. The idea is precision. And so the ability to do precision combinatorial treatments is really, really very good. And there are a lot of detailed things that I don't have time for, where we developed the tear and all, and showed how effective it is. And do I have another 5 minutes? Otherwise, I can stop here and not get into the regenerative medicine.
Yeah, maybe you can stop it here. Yeah.
Okay, fine. So I was going to talk about regenerative medicine, which is a whole big new field of nanomedicine. Because when you have tissues that are failing or that are need repair, you have arthritis, and you have so many other conditions, the bone loss, and even heart tissue and so on. Regenerating is extremely attractive with nanomaterials. It's a tremendous amount of work that has been done. We have done some good work, but it's a phenomenally good area. So I'm going to skip it and come to the conclusion so that we can move forward. I wasn't sure if I would get to the regenerative medicine part, but I kept it there in case there was interest. So you can see there's a lot of very interesting work. So if you look at the important points, just to quickly summarize, You can do targeted drug delivery, photodynamic therapy, an oral drug delivery platform, diagnostics, and also, as I said, metronomic therapy is another very interesting area which is intriguing and needs to be addressed. Combinatorial therapy, where you can combine things, as well as theranostics, which is both diagnostics and therapy at the same time. And those are potential and abilities which cannot really be done any other way very well. And I talked about tissue engineering, but we didn't have the time to review that. Then immunotherapy, the future, I think, if you're looking at it, is the inroads that nanomedicine is going to make in immunotherapy, cell therapy, gene therapy, vaccines, and so on. We are looking at TB vaccines, we're looking at cancer vaccines, and gene editing, are all areas where there's a huge potential and application for nanomaterials. And worldwide it is being done. It's not the new idea that I'm saying, but there is nothing really out there that is really coming out, but there's a lot of active work that is going on. In regenerative medicine, I think there is some genuine talk about organ regeneration, particularly when it comes to skin and maybe liver, which has potential using nanoscaffolds and so on. Finally, I'll end with my thanks to Department of Science and Technology that has made a huge contribution to us and to India in terms of supporting the role of nanomedicines in medical applications, and also Department of Biotechnology for their support. And of course, our Chancellor Rama has invested a lot of funding and support in labs, equipments, and personnel to help drive this effort. I apologize for the delay in starting it. I didn't anticipate that I'll have so much trouble getting on with my talk. So if there's time for some questions or comments, I'll be more than happy to discuss it. Thank you very, very much for this opportunity.
Thank you very much, Professor Nair. Thank you very much for a very nice introduction with the overview, and as well as different example with the translational and application point of view. And there are so many things that I see, I think, yeah, so the stage is open for questions. Yeah, if you have any question, please, you can unmute and you can directly ask, or you can write in the chat box. Is there any question?
Good evening, sir.
Yes, please go ahead.
Hello, sir. Sir, yes. Myself, Pasneha Bandekar from.
I think you're on the street somewhere.
Am I audible, sir? No, sir. Actually, it is a track is beside my house only.
Okay.
The train is going on. Okay. Sorry, sir. Sorry for inconvenience.
No problem. Go ahead.
My question is, we are synthesizing nanomaterials.
Sure.
Yes. So we are synthesizing silver nanomaterial from plant extracts, different plant extracts.
Sure.
And we are testing the antibacterial also, sir. Means whether it is fighting the bacteria or not, we are tested that also.
Sure.
The results are really good, sir. But if in further, sir, in the further scope, if you want to apply for the nanomedicine, means coating for medicines and all, so this toxicity and efficacy, how that can be tested, sir, for our the samples, synthesized nanomaterial?
Well, efficacy can be tested with so many, methods, in vitro as well as in vivo, for antibacterial effects and so on. Toxicity is more of a regulatory thing, the absence of any antibiotics or any drugs that are used.
Efficacy and toxicity can be tested, no, sir? So because we are from engineering background,
it's pretty standard.
We don't have that facility here in our college. That is my question, sir. If you want to go for further studies, application of the synthesized materials in the medicine, nanomedicine, at least for the coating, because
Yeah,
we
toxic,
getting good results antibacterial test
yeah.
and all.
Toxicity is something that should be done in generally in animal studies, preclinical studies, so that you know that there are no unwanted side effects or other problems that you see. So it requires basically an animal lab, small or medium animal labs. Normally, before you translate anything to the clinic, they will want a large animal regulatory tox study as well, so that it can be approved by regulatory bodies. So if you're thinking about actually translating something, then there is quite a process involved, as it should be, before it can be approved.
Okay, I think we can.
Okay,
move
sir.
on.
Thank you.
Yeah.
Okay.
Yeah. Is there any? Hello, is there any query or question? Please unmute yourself. We can ask. Yeah, while someone is asking, sir, I have one query about this this nano medicine efficacy regarding this toxicity. So you showed a curve that's showing that toxicity and efficacy is actually inversely sort of they're going in for nano medicine. So in that curve, what is the parameter do you change, for example, to make it efficacy higher but toxicity is lower? Is it the size or safe, or I mean, microscopically, what do you change of
No,
nano?
one example of that is targeting. If you add a targeting ligand, you increase the efficacy, but you reduce the toxicity because it doesn't go elsewhere and do other damage. So it really depends upon what parameters you're controlling, but you have the ability to increase the efficacy and decrease the toxicity, which is not something that you can do with conventional medicine.
Yeah, it's a non-intuitive. Yeah, absolutely.
Yeah, it's not as obvious, that something like that can be done. And these are the reasons why I think there was a lot of interest in these non-intuitive sort of results, which people have found.
Yeah, absolutely. Yeah. Okay, thank you very much, sir. Okay, we are running out of time. I hope there are no more questions. So Venkatesh, I'll hand over to you to conclude this today's talk.
Good evening to everyone. We wish to express our sincere thanks to Dr. Shantikumar Nair, Dean and Director of Amrita School of Nanosciences and Nanomedicine, Amrita Vishwa Vidyapeetham, Kochi, for his excellent talk. And thank you very much, sir.
Thank you.
One special thanks to Professor Anindya Das, convener of Bangalore India Nanotalks 2022, and also as a professor, Department of Physics, Indian Institute of Science, for his valuable guidance in planning the Bangalore India Nanotalks. Thank you, sir.
Thank you very much.
I also thank all the participants of this program. Thank you very much for one and all again. Thank you, sir.
Yeah, good. Yeah, good evening to everyone. I think, yeah, good night to everyone. Thank you very much. See you, sir. Okay.
Yes.
Bye, sir. Bye, Nair, sir.
Bye, sir.
Yeah.
Bye, sir.
Thank you.
Thank you.
Thank you. Shall I sign out?
Yeah, you can leave. Yes.
Okay, I'll have to unshare or something like that.
I think you can if you otherwise you may not see the leave button. Yeah.
Yeah.
Okay.